Hey team, welcome back to Conservation Genetics. Today's lecture is the start of the big game. So you've got the basics, now we're going to talk about genetic diversity in small populations. This is where it starts getting really interesting. Outcomes of this lecture, I want you to understand what the populations of conservation concern are, the idea of an idealized population or effective population size is really important, and you need to understand how we get it and why we have it. We're going to look at genetic drift, we're going to look at genetic bottlenecks, we're going to look at selections in small population. This is where the rubber hits the road in terms of conservation. So again, you guys have been doing really good with the questions. Keep questions coming in. And I do appreciate the one student who keeps telling me the course is too easy. And I do my best to make sure you're not bored for the rest of the semester. Why do we care? Obviously we care about small populations because they're the populations of conservation concern. Those are the ones that we need to fix for conservation, right? They all have small or declining populations, and obviously these populations are more prone to extinction. All of these guys have small or declining population sizes. These are the things we're trying to keep alive right now. This is the big game for most of us. For most of you guys, this is what you're going to spend a lot of your career working on if you decide to go into conservation as a field. It's going to be keeping these species alive and doing one better than that, contributing to their recovery. To do that, you need to understand how they work when they're small. So we're going to go back to the IUCN Red List. It was established in the 90s. It's the global standard for assessing extinction risk and evaluating conservation status of both plant and animal species, and they consult with scientists who have the best knowledge of the species to assign a conservation status. Those statuses could be anything from, so there's a lot of species that are not evaluated. Don't think this is a field where we understand everything. And if they are evaluated, they can just not have enough data. That's data deficient. Then it's everywhere from least concern up to critically endangered or extinct in the wild, which means there's still some populations in captivity or just extinct overall. Remember that the threatened categories and the extinct in the wild categories come with legal protections. So there's a real benefit to using this system because it stops the trade in species and it stops what people can do with them. However, and we've talked about this before, how you classify a population as critically endangered, endangered or vulnerable has to do with the actual or projected population size decline, the extent of occurrence of where the individuals live, population numbering, and the quantitative analysis showing probability of extinction in the wild. Now, besides the last one, the other ones have nothing to do with genetic diversity. And the population numbers here are interesting. So critically endangered is less than 250 mature individuals, endangered is less than 2,500, and vulnerable is less than 10,000. So what we're saying is anything with more than 10,000 individuals is not vulnerable, we don't have to worry about them. And that may not be true. And that sounds crazy. But we're going to have a look at the genetic size of populations as we move through the second half of the course, and we're going to see that just like with the northern elephant seal, you can sometimes have a large number of individuals that are not in good shape genetically. So in a small population you have genetic costs. These are the Mauritian kestrel. They're a really cool bird, but they have small population size. Small population leads to higher inbreeding, lower genetic diversity, reduced reproductive fitness, compromised evolutionary potential. It's hard for them to stay alive, it's hard for them to evolve with a changing environment, it's hard for their population to recover. They've got lower fertility than other species of kestrel that are closely related to them, and higher adult mortality. Lower productivity, they make less eggs. So they did a genetic analysis on them, and you can see the allelic diversity is 1.4, and their heterozygosity, the expected heterozygosity, which is a really good measure of just easy measure of genetic diversity is 0.1. Ancestrally, it was more than double that, and the allelic diversity was 3 times what it is now. If we have a look at the non-endangered species that they're closely related to, you can see heterozygosity is typically about 6 times what we find in these guys, and the allelic diversity is 4 to 5 times what we find in these guys. There's a huge difference between the endangered ones and the non-endangered ones in terms of their DNA. These guys don't have a lot of variation. The non-endangered ones do, and that contributes to the fact that they find it harder to survive and to reproduce. Now, evolution works different in small and large populations. Remember we talked about the things that contribute to evolution, so the change of populations over time. There's selection, mutation, migration, and genetic drift. And in large populations, selection, mutation, migration determine the outcomes. These are the most important things. That's what it looks like when it's working. In really large populations, over large amounts of time, mutation is actually very powerful. It doesn't do anything short term, but over long periods of time, it's powerful. Selection is obviously powerful, and it's the one we want to be working, and migration is important because it determines how species move around. In small populations, these effects are less powerful, and what becomes more powerful is genetic drift. Genetic drift is a chance effect. The smaller your population gets, the more important chance gets, and that's problematic. Okay, so census size of a population. This is what the IUCN measures. It's called N. Census size doesn't tell you a whole lot about the composition of a population. Just like your weight on the scales doesn't tell you a whole lot about the composition of your body. So if you wanted to look at the composition of the body, you would look at things like body fat percentage, right? If you want to look at the composition of the population, you need to look at more than just the census size. Census size doesn't account to reproductive size because it doesn't understand fluctuations in size over time, doesn't look at juvenile and post-reproductive individuals. Obviously, they don't contribute in the same way because they're no longer able to create offspring. Doesn't look at sterile individuals, doesn't look at variation in reproductive success. That's really important. In a lot of lekking species of birds, they're birds where the males dance or less. 2 or 3 of the males will get the vast majority of the mating, and most of the males will get none. That's going to heavily impact genetic diversity. Doesn't look at the breeding system. The breeding system is really important. If you have species like Cunningham skink, which unlike a lot of bird species, these are lizards that are truly monogamous, genetically monogamous, most of the time for their entire life. So if you're another male or another female, and there's 3 of you, your genetic material is never going to be passed on. And then population structure. Census size doesn't look at population structure. So none of these things are considered in census size. That makes it really hard to compare a group of antelope to a group of ants, to a group of Mauritian kestrels, because all of those things will be very different between species. So we do something instead. We create the idealized population. In an idealized population, all of your adults are hermaphrodites that randomly mate. The number of breeding individuals is constant over time. There's non-overlapping generations. It's a closed population with no migration. There's no selection and no mutation. And the number of offspring per adult is 1 with a variance of 1. So the population is stable. It's not growing or getting smaller. Now, the idealized population, much like Disney hair, is not real. It's not intended to be real. It's intended to allow us to compare a population of antelopes to a population of ants. Because if we can figure out how big that population of antelopes would be at an idealized population, we can figure out how big the population of ants would be at an idealized population, then we have a number we can compare across species. And that lets us build rules about how many individuals you need in your idealized population to be stable in the short term, to not have a lot of inbreeding depression, and to be stable in the long term to retain evolutionary potential. This term is NE, or the effective population size. So it's the size of an idealized population would lose genetic diversity at the same rate as the actual population. NE is almost always smaller than N. Um, in fact, in practical terms, it's always small on n. It accounts for factors that reduce the capacity of the population to retain genetic diversity. So it accounts for non-random mating, it accounts for deviation from a 1-to-1 sex ratio, variation in family size, fluctuations in population size, overlapping generation, and inbreeding. All of those things contribute to the NE, or the effective size. So if you can calculate the effective size of a population, you'll have an idea of how it's really doing genetically. It's a really good measure. And so they went out and did it. They went out and did it to 192 different species. 102 different species, 192 estimates. To see how big the effective size was compared to the census size. And the number they found is a little bit horrifying. Ratio of NE to N is about 0.11. What that means is the effective size is usually around 11% of the census size. That's a concern. If you have 100 individuals census size, your effective size is about 11. If you have 200 individuals, it's somewhere around 22. That's a problem. Because this effective size tells us how a population is doing in terms of their evolutionary potential, in terms of their ability to avoid inbreeding depression. And they're a lot smaller than census size. And remember that the IUCN looks at census size, not effective size. So what are the advantages of the IUCN approach to classification? What are the main genetic costs associated with small populations? How does ploidy affect these costs? That's a really good question. What is the general trend in the relationship between effective size and census size? And just for fun, I'm going to put one of these questions in your mid-semester test. So go in and think about these and make sure you know how to answer them. Potentially use the discussion forum. Next thing we're going to look at is genetic drift. Genetic drift is random chance, and just like happened to this yacht, random chance is not always a good thing for a population. So here we're looking at a couple of cheetahs. So we've got 4 cheetahs. They have 3 different alleles, A1, A2, A3, at one locus. But when you have a look at their offspring, the A3 allele is missing, just by chance. So only this individual had it, and she passed on her A1 allele, not her A3 allele, and these two only had one offspring. In this case, frequency of R, which is the A3 allele, has gone down to 0. It wasn't selection that did this. This allele didn't mutate away. Just by chance wasn't passed on, because all diploid individuals have 2 copies of each allele, and they only pass on 1 at a time. Another way to look at this over time is it's just these stochastic populations. So we start off with 50-50 green and yellow beads, and we draw out 10. That 10 become the basis of the new 100. So just by chance we draw out 6 yellow and 4 green, and I actually used to get you guys to do this in class, so thankfully I found a diagram that shows it just as well. Now we redraw. Now the next time, by chance, we draw out 7 yellow and 3 green. So now it's 70-30, because we use those same 10 to restock. So 10 we made into 100, and then this time by chance we draw out 4 yellow and 6 green, so the change is 40-60. Now in a large population, these changes are very small, because you're picking a lot of individuals out from a really large bag, and statistics tells you that you're going to end up with changes, but they're going to be relatively small changes. The smaller your population gets, the bigger these swings can be. So what we can see here is that we kept restocking. So we went 6 and 4, we turned that into 60 and 40, and then by chance we drew out 8 yellow and 2 green. And that was restocked to 80-20. And then by chance, we drew out 10 yellow. And as soon as we do that, the green are gone, because we restocked to 100% yellow. That entire allele is lost from the population. Now, that's how it works in a large population of 100 beads, but when we have a small population of only 20, these swings happen a lot faster. Doesn't mean you're definitely going to lose the green or definitely going to lose the yellow, but the chance that you're going to lose one or the other is higher. So genetic drift is more powerful in small populations. If this doesn't make sense to you, honestly, go buy a bunch of M&Ms and try it. In small populations, genetic drift leads to the fixation of alleles. What that means is, where there was 2, now there is 1. One of the alleles is lost completely, the other one is at 100%. And the way that it picks which allele to bring to 100%, which one to 0, is completely random. So it diversifies among replicate populations. What that means is sometimes you'll lose the allele and sometimes you won't. Sometimes it'll completely disappear, sometimes it'll be 100%. The effect of genetic drift is even stronger on rare alleles because they're the most easily lost. And really importantly, in small populations, genetic drift can overpower selection. So what you can see here is some simulations. Here we had an effective size of 2 individuals. Here we had an effective size of 10. We had a couple of generations. We started off with allele frequency of 0.5, so 50% big A and 50% little a. And every time it hit the top or the bottom, it meant the simulation said that one of the alleles was lost. So you can see that in all of our runs, 1, 2, 3, 4, 5, 6 times, the allele is lost. And only in 4 cases is the allele not lost. Now when the effective size gets a little bit larger, we've only got a couple of generations here, but you can see that all cases, in this run that we did, in all cases we kept both alleles. We didn't lose one. Alright, this is the bit we've touched on a couple times. In small populations, genetic drift and selection compete, and alleles that have an effect on fitness might behave as though they're neutral because the effect of resampling or chance is so high. So anytime your selective coefficient, we talked about that in our last tutorial, is less than 1 over 2 NE, the allele will behave as though it's neutral. What that means is things that are mildly to moderately deleterious, so they're bad for you, they're not lethal, but they're bad for you, they start to act like neutral alleles. And neutral alleles drift. They move around by random chance. Neutral alleles, because they're drifting, they can become fixed in the population, which means they can become 100% all of the alleles. So in small populations, selection is hamstrung to such an extent that deleterious alleles that would normally be removed from the population or maintained at low levels through mutation selection balance actually become the only allele at a locus. That's really bad. That's why Godzilla is facepalming. Also, I just like this picture. If you want to look that up, it's called, that effect leads to something called Muller's ratchet or mutational meltdown. So, which alleles are affected most by genetic drift? You should be able to figure this out. What's the relationship between drift, selection, and population size? The deer population, this is a real thing, of St Andrews estimated 180. That led UNB students to suggest a cull. They were like, well, there's 180 deer near St Andrews University, so us as environmental managers suggest that we should cull them because there's too many deer. Think about that as a geneticist. Is 180 a big enough number to start to cull? The next thing we're going to touch on is population bottleneck, and this is where you have really good genetic diversity until something happens that narrows the amount of individuals that survive. So here only 4 survived. 4 alleles survived. Small number of individuals. Could have been 2, could have been 10 individuals, but only 4 alleles survived. And that means that all of the diversity that was present up here is now gone. And remember from our mutation rate stuff, this is going to take 100,000 generations to come back. It's a big problem. Okay, here we're just going to model some genetic sampling. So the chance of getting any one of these four outcomes is 25%, which means that the chance that one allele is lost, either A1 or A2, is 50% if only one offspring is produced. So if these guys only made one offspring, then if the offspring is this offspring or this offspring, they're going to lose one allele. That's how drift works. That means if we extend this a little bit, we assume two gametes are sampled per offspring, which makes sense because offspring are made of one from one parent, one from the other parent. If there is n number of offspring, the chance that an allele will be lost is half to the power of 2n. Obviously, the larger n is, the less likely it is that an allele will be lost. But the chance that we'll lose an allele in any given population is half to the power of 2n, assuming 50%. So A1 and A2 at 50% each. However, we don't typically have 50% each. We have P and Q because alleles can have any percentage. Yeah, it could be 80% of the population, it could be 10% of the population. Obviously, the alleles that started off rare, we're more likely to lose. And so we have an equation to find out our probability of losing A1. Where A1 is any allele. And it's 1 minus the frequency of that allele to the power of 2n. So you try this a couple of times. Try it with a rare allele, where P equals 0.01. Try it with a common allele, where P equals 0.04. 0.4, not 0.04. So if you make P equals 0.01, you're saying only 1% of the alleles are P. And if you make it equal to 0.4, then 40% of the alleles are P. And pick a small population, say 5 individuals. And then pick a large population, say 200 individuals. And see what happens to the chances of losing that allele. And that's exactly what I want you to do in the next question. I knew I wrote that for a reason. Give it a shot, see what happens. And the answers are here, and they are kind of what you expect. The probability of losing the allele that is relatively rare is about 10%. The probability of losing the allele that's common is much, much, much lower. And if you were to manipulate the population size, so if you were to change population size of 4 to 40, obviously it's going to have a big effect as well. So a couple more. Now that you've got the example down, we're going to have a look at a population of teddy bears. And we'll look at two different loci. The first one is in part 1. 2 alleles where the probability of the first one is 0.4. Now obviously P plus Q equals 1, right? You should be able to figure out the probability of the second one. What is the chance that the first allele is lost? And a second loci in the same population has 3 alleles, P, Q, and R. Find the probability that any one of them is lost. Not just P, not just Q, but any one of them is lost. Given that we're down to 6 individuals. Okay, so what is a population bottleneck? We know they're bad. It's just any sharp reduction in population size. And it means one good thing has happened at least, okay? So for there to be a reduction and then for it to come out again like this, obviously means the population is getting larger, which is good. It's not just continuing down into oblivion. It's not just going extinct. The population is getting larger, but something caused it to contract. It can be overexploitation. For a lot of species, it was loose hunting regulations, and so we overhunted them. In a lot of fisheries, this is exactly what's happening to about a third of our fisheries. That's between a third and 2/3, to be honest. So 33 and 66%. Some people say 80% of all fisheries on the planet are overfished. It's leading to bottlenecks in genetic diversity. In some cases, we're now fishing areas of the ocean that we really shouldn't touch. So we're fishing, for those of you who did my first year unit, we're fishing down into the bathyl pelagic, into the deep, cold areas of the ocean where there is no light and very little energy, and species of fish that live down there take a huge amount of time to reproduce and grow. And so we're pulling up these little fish that could be 40 years old, 50 years old. Their populations can't handle the level of harvesting that we do with the net. It causes massive problems. So the census size recovers. In some cases, if you control, you got to get in early, right? You can't do what we did with the Newfoundland cod. If you get in early, the census size will recover. You put in regulation, you stop the over-harvesting, you clean up the pollution, you've done something really good. But the genetic diversity is still gone. That's problematic. I've talked about the northern elephant seal, so we might talk about them in a little bit more detail. There's a bunch of good papers on this if you want to look it up. Hunting caused the population to decline to 20 to 30 individuals. There was only one rock left with these things on them. Hunting was then banned because that's a shitty thing to do to a species. And they have recovered to 175,000 individuals, which sounds amazing. It sounds like we won in conservation. But when we look at that 175,000 individuals, we only have 2 mitochondrial haplotypes. So a minimum of 2 females are represented. The heterozygosity is 0.16. In a species of closely related seal, the heterozygosity is 0.875. So it's 8 times as large, or 7 times as large amount of genetic diversity. Now, we got this population back to 175,000, which is amazing. But how well are they going to cope with the warmer waters that we're getting due to climate change? We're not sure. How well are they going to cope with new variants of disease? Remember, these guys are mammals, minks are mammals, minks get COVID, these guys might get COVID. How well are they going to cope with that? Given they have very little genetic diversity. So even though we brought the population back up to 175,000, and by all of the measures of census size, this population is fine. If we look at their genetic diversity, we see a different story. Okay, so if we look at the genetic diversity, I would still recommend that we manage this population, that we look really closely for diseases, that we make sure that we've got seals located in different geographic areas. Because if a disease takes out one seal in one of these colonies, it's likely to take out many seals. So it should be a priority to make sure we have different colonies of seals on different islands. Even though we have over 100,000 of them, almost 200,000. The next experiment, which I've talked about a little bit, is the Steve O'Brien experiment. He looked at 55 cheetahs and found that he tested them for 47 different loci and found no genetic variation at all. That led him to think that these guys suffered a bottleneck sometime in their past. So this is we're not sure when. It's old. At least 10,000 years old, I believe, was his conclusion. So in that 10,000 years, cheetah population has not recovered a significant amount of genetic diversity. He wanted to prove it. So he took a skin graft from one cheetah and slapped it on another cheetah. Now, if we did that in a human, your body would fight that skin graft. It's why if somebody has a lung transplant or a heart transplant, they need to be taking immunosuppressants for the rest of their life, because their body will reject the organ. Cheetahs don't reject the skin. There's no immune effect at all, because they have such little genetic diversity that they can't tell the difference between their body and the body of another cheetah. That's a concern. They've repeated this study a couple of times, most recently in 2011, and found a little bit higher MHC in wild populations than zoo populations, which is good, but still pretty garbage. And MHC is just an area of the genome that controls immune response. If you want to freak yourself out, why would you not want to do this? Look up the Wiedekind t-shirt experiments. That was having a look at how MHC controls mate choice in humans. Also works on a bunch of other mammals and a bunch of other vertebrates, to be fair. Again, they did a lot of experimental work on population bottlenecks, particularly in fruit flies, because they're easy to study, and found exactly what you expect, that we have an outbred population with lots of different alleles. When we do population bottlenecks and then bring the populations back to being large, they lose a significant number of those alleles. And that when you have a population that bottlenecks multiple times, so it gets small, then it gets big, then it gets small, then it gets big, the genetic diversity loss is cumulative, obviously. if they went down to a single pair, they lost 25% of the heterozygosity immediately. In fact, we have an equation that I want you guys to get good at. This is one of the ones I'm going to ask you about a lot, where we look at the proportion of heterozygosity retained from a single generation bottleneck. So H1 is the heterozygosity after the bottleneck, and H0 is the heterozygosity before the bottleneck. So the ratio of heterozygosity lost is 1 minus 1 over 2n, where n is the size of the bottleneck. That's actually, it means that you can bring a population down quite sharply for a single generation, and you're still going to retain a significant amount of genetic diversity. So one generation bottleneck is bad, but you can deal with it. It's when the bottleneck starts to go for several generations that the effect compounds and loss of heterozygosity gets worse and worse. Which means that just like everything in conservation, we need to use this to find a positive. If a species is bottlenecking really quickly, we need to turn that around as fast as we can and get it growing again. Because a single generation bottleneck will lose 25% of the genetic diversity if it gets down to a single pair, but you still have a significant amount of genetic diversity. But you have to get it back up to a reasonable size within the next generation. What we've learned from this is loss of genetic diversity is predominantly from sustained reduction in population size, bottlenecks that last multiple generations. And the effect on allelic diversity, which remember the number of alleles, is more than the effect of the effect on allelic diversity is greater than the effect on heterozygosity. It's because allelic diversity looks at how many different alleles you have, and rare alleles are the most likely to be lost. And you're going to lose alleles before you lose heterozygosity. So again, equation for that, number of alleles retained equals n minus sigma 1 minus pi to the power of 2n. Have a look at example 8.2 in the book. It shows you how to do this. Again, this is something I'll test you on. We're going to move on and have a look at founder effect. Founder effect is a type of bottleneck, but not necessarily because your initial population got small, because a number of individuals broke off and started a new population. Think of the people who decided to walk north and ended up in Japan and founded Japan. Think about the couple of birds that fly a little bit too far and end up on a new island and decide to stay there. So it's when a large population is established by a very small population. Population in Japan is big now, but there wasn't 300,000 people who walked to that area. And we have an example here. Here you have red and blue, and we have 3 populations that were founded by some individuals from this population. In one case, only red alleles came over just by chance. In one case, only blue alleles. In one case, both alleles. And so if this population gets massive, let's say it gets to 500,000, it's still only going to have that red allele. Because just by chance, nobody with a blue allele came over. A good example of this is bighorn sheep. We use bighorn sheep as an example often because we know a little bit about their genetics and they're relatively easy to study. So one of the island populations started about 20 individuals. It's now up to 650 individuals, and because of that, because they were like, oh look, this is a healthy population with lots and lots of sheep, they used them for translocations. They brought them into northern Mexico. But when they looked at the genetic diversity of this population, it was low. So they were supplementing the northern Mexico population with individuals with low amounts of genetic diversity. They weren't adding anything back in there. It would have been better to take, or potentially better to take, individuals to supplement the northern Mexico population from some of the other nearby populations because they were in better shape genetically. Just because the census size is big doesn't mean it's got good genetic diversity to back it up. Also means that if I was managing this species, I'd look at the population on Tiburon Island and I'd try to bring some individuals in to reintroduce the genetic diversity that got lost when they only started with 20. So I'd bring some in now because that would have a huge effect on how healthy the island would be going forward. Now every time I teach this, somebody says But what about invasive species? Invasive species have low founder numbers, and they can be problematic. And we have starfish. There's a type of crab in Prince Edward Island that's an invasive crab, and it's problematic. Probably did start with a few individuals. In Australia, we know exactly how many rabbits were initially brought into the country, and now there's just a gigantic number of the stupid things. So how does this work if their genetics isn't very good? Well, firstly, when we look at invasive species, we have ascertainment bias. So somebody in Charlottetown won the lotto a week ago, and everybody ran out to buy lotto tickets, right? Which is dumb, because nobody looked at all of the people who play the lotto consistently and lose. They just looked at the winner. They couldn't see the invisible data. The invisible data in invasive species is, yes, the green crab is invasive in PI, but many other species of crustacean came in on those ships, and those just died out. We just didn't see those. The other thing to remember about invasive species is they're in a new environment where selective pressure is relaxed on them. They don't have predators and they don't have diseases they're used to. In those cases, if you have no predators and there are no diseases that affect you, your population can grow despite having really poor genetic diversity. Okay, a couple more questions for you guys to practice on for the next tutorial. And then just a quick summary. I know some of you guys have said you're not interested in these. There's nothing more in this lecture. You can turn it off now. If you're sticking around, I want you to understand what the IUCN does and what they measure to get their standards. And then I want you to understand that effective size is typically much lower than census size, which means that populations are more vulnerable than their census size suggests they should be. How much more vulnerable? We can talk about them in a later lecture. In small populations, I need you to know that genetic drift is really important. Unlike in large populations where it has a small effect. I also need you to know that in smaller populations, genetic drift can overwhelm selection. It can make selection not work. And then you need to know how population bottleneck works and the effects of population bottlenecks, and that founder effects are exactly like population bottlenecks. So even if the source population is doing well genetically, sometimes some of the island populations are not doing well because only a small number of individuals founded those populations. And that is it for this lecture. Thanks very much, guys. Take care.